you. How's everybody doing?
Good.
Excellent. All righty. So, yes, my topic is talking about the brief history of CRISPR and Cas9 research and development. This is a big topic, and so I'm going to try to compress as much as I can into 20 minutes. But before I begin, I do have to ask two questions. Just by a show of hands, who has heard of gene editing or genomic editing before? Okay, now for the tougher question, if I were asked you to define it, do you think you could do a good job of defining what gene editing is? Okay, I got a couple of people. So, in order for us to be on the same page, I'm using the Merriam-Webster's dictionary definition of gene editing, and it says is the use of biological techniques to make changes to specific DNA sequences in the genome of a living organism. And for all intents and purposes, the biological technique that we're going to be looking at is CRISPR-Cas9. Probably the biggest revolution, and the way Dr. Eric Lander from MIT describes it, it's hard to recall a revolution that has swept biology more swiftly than CRISPR has. But before we get into the research, I do want to briefly touch on the structure of scientific revolutions. This is a book written by Thomas Kuhn in 1962, and basically what it revolves around is how scientific thought that is ultimately revolutionary comes into being. And it goes around four major points that he says. First one is science is driven by curiosity and discovery. We look out into science, we see something, and we see order to it, we see its purpose, and we wonder why it's there. And so from that, we are able to form these general paradigms or core beliefs such as the law of gravity, such as the central dogma of biology that states that DNA is transcribed into RNA, which is then translated into protein. And from these, we make bigger conclusions, we draw on new inferences and new references. And the research that we dive into ultimately strengthens our existing paradigms, or what it does is it introduces new ones. And from this, we ultimately get what are known as crises, as Kuhn calls them. They are the kind of the birthplaces of scientific revolution, where two theories are in contrast to one another, or two things that we had thought were commonly believed and held to that are now in conflict with one another. And what I propose is that CRISPR-Cas9 is no different than any other scientific revolution, no different from Copernicus moving Earth from the center of the universe to around orbiting the sun. It is just as fundamental, but not different. And I would say that as we look through the research that has been concluded, we needed essential paradigms to build us up to the point where we are now utilizing CRISPR-Cas9 as a gene editing technique. So, early CRISPR research, it really began with kind of an unexpected discovery in 1987. A group of Japanese scientists were looking at a gene in E. coli and noticed these 5 palindromic repeats on the end of the gene. And they didn't really think anything of it. They were really focused on the gene that they were looking at, but they did mention in their article these novel type of repeats that they had no idea what their purpose was, but they were there. And to be honest, scientists could have dismissed this as junk DNA, just DNA that had been left over from another organism or from another protein. But in 1989, Francisco Mojica at the University of Alicante along the eastern coast of Spain was studying archaea, which is a microorganism that is similar to bacteria but ultimately biochemically different. And what he found were these same types of repeats within the genomes of the archaic bacteria or archaic organisms that he was studying. And from that, he was ultimately determining and kind of coming to his own conclusion that if it's in archaic microorganisms and in bacteria, there's got to be some reason for them. So he began sequencing them, taking them out, and kind of studying them a little bit more. And by 2000, the year 2000, he had found 20 other microorganisms that displayed these sequences. So this was starting to become a little bit more than just a new novelty. So in 2002, Rod Jansen, building on the research that Mojica had done, decided to have his own study. He did an in silico study going through all of the known genomes and databases that he had of microorganisms, especially prokaryotes. And what he had found is that between 40 and 50% of bacteria and over 90% of archaea actually displayed these sequences. So these aren't just novel, They're found almost ubiquitously throughout nature and found interspaced around. And from this, we were starting to get the idea that there's a little bit more here. So Mojica actually did his own in silico study of these small repeats, or as he had now coined them, clustered regularly interspaced palindromic repeats, or CRISPR. He's kind of the father of CRISPR research. And what he did is he wasn't looking at the tiny repeats anymore. He was now looking at the DNA in between the repeats. And what he did was he did a search through the genomes of this DNA in between, and what he found was that it actually was a match of a virus known as P1. So why would bacteria have this viral DNA incorporated into its genome was his question. And ultimately this would help us finally figure out where we were going with it. But in 2005, while studying these CRISPR sequences as well, Alexander Bolton actually found the discovery of proteins that were closely related or close to these CRISPR sequences. And so now we have all the building blocks, in my opinion, to now start to make scientific conclusions. We have proteins that are by these repeats that include viral DNA. How do they all work together? So the discovery of CRISPR's purpose started to take a little bit of shape in 2005, where Banguru, Horvath, and Moyu at the dairy giant Danesco actually started to do some research. And what they were looking at is looking to increase yogurt production by studying the probiotic Streptococcus thermophilus. And what this probiotic does is actually give us the ability to create yogurt from milk. And so what they noticed in these bacteria is that the number of phage sequences that were incorporated into the CRISPR DNA ultimately gave the microbes an increased immunity to resist viral infection, which allowed them to then have better increased milk production or better increased yogurt production. And so this was kind of a first real confirmation that this was actually doing something within the microorganisms. The first identification that this was a sort of adaptive immune system that the bacteria were using to repel viral infection. And they would later go on to determine more Cas proteins, their function, and ultimately the CRISPR RNA that matches up with Cas9 proteins that we will get to later. But later that year, John van der Oos and Eugene Kuhn discovered CRISPR systems. So they discovered the purpose behind the Cas proteins and discovered two different types. A cascade that involved various CRISPR proteins to ultimately chop up the DNA of any virus that was infecting them, and type 2, which only included just one CRISPR protein, or Cas9. And this would be important because as they continued to look into it, Cas9 started to introduce what was known as double-stranded breaks, where it cut the DNA in two, giving you two separate strands of DNA. And where we would go with this is starting to indicate that CRISPR-Cas9 ultimately is the adaptive immune system. But in 2006, Emmanuelle Charpentier offered a kind of a new wrinkle to it. She introduced what was known as tracer RNA, or the RNA that was created off of the DNA in between the CRISPR repeats. And this RNA would ultimately serve as the precursor to the CRISPR RNA that gets matched up with Cas9. And without this discovery, it would have been almost impossible to bring together the two pieces. And from that, but there was one other piece that we needed to get to, and how was this operating? Was it attacking RNA that was being created by the viruses integrating themselves into the DNA, or was it actually targeting the DNA when it was injected into the virus? And so Lucio Marafini and Eric Staudenheimer performed a very interesting experiment using a self-splicing intron. They put this intron into the CRISPR array and then had it be translated or transcribed through, and once it was transcribed through, it would cut itself out. And so by doing that, they found that when the intron spliced itself out, it wasn't cut up, it wasn't at all hit, neither was the messenger RNA that it was creating, indicating that CRISPR actually cut the DNA that was implanted by the viruses. And so from here, we are able to get the big breakthrough that happens in 2011 and 2012 from Jennifer Doudna and Emmanuelle Charpentier's partnership. Jennifer Doudna working with her research group at the UC Berkeley, and Emmanuel Charpentier working at the University of Umeå in Sweden. And from this, they correctly deduced that Cas9 was the protein involved with cutting up the DNA. And so what they needed to do was first extract the protein from E. coli, purify it, and then start to perform some experimentation. And rarely does science ever go the way we want it to. And so the first time they did this, it didn't work at all. Because they were missing a key component with the CRISPR RNA. They thought the protein was enough and it didn't quite cut it. And so after this, Martin Janek and Christoph Trulinski's experiment ultimately included the RNA that it needed, the CRISPR RNA and the tracer RNA, and the Cas9 protein, along with the correct matching DNA sequence. And that was the first kind of programmability that was able to display with CRISPR-Cas9. It highlighted the necessary components that we needed to ultimately have this experiment. So from all of this that we have the Cas9 protein that is involved with the cutting. We have the target DNA that matches up with the CRISPR RNA, or the guide RNA as you see there, and that matches up specifically by Watson and Crick base pairing. So it is extremely high in specificity and allows it to create these breaks. It will only cut if it matches up correctly. And Doudna and Charpentier actually were the first to show this, and then it gives us the question of, is this only regulated to prokaryotic organisms, or is this up to higher eukaryotic organisms? So in 2013 was kind of the big, overwhelming rush of experimentation. We have 4 different papers on human editing, or editing in human cells, and 1 paper regarding embryo editing, specifically zebrafish, allowing the researchers to create the model organism that they wanted to study. And this was huge. This showed that this was not just a prokaryotic mechanism that we could use. We can now use this in higher organisms moving forward. And this has played very big into the publication, or the patent battle between all of these competing scientists as to who came up with this technology first. Ultimately, right now, as of last month, UC Berkeley has been awarded the patent tentatively, but that might be subject to change over the next couple of years. So, with that being said, with the creation, now that we have this protein, there have been improvements that have been made. One of the big problems with CRISPR-Cas9 is what's known as off-target mutations, where all of a sudden the sequence of the DNA matches up with the CRISPR RNA way too similarly, and so it starts to cut in places that you don't want it. But we've been able to create higher fidelity CRISPR RNA, which allows us to remove those off-target effects. And we've also been able to introduce new CRISPR proteins and determine their interactions, to not just create double-stranded breaks, but to also create what are known as sticky ends, or overhand breaks, which allows you to insert genes into the genome. But one of the other big problems is that this is a protein, and it doesn't pass through the cell wall very easily. And so one of the biggest things that has been done in its improvement is the delivery mechanisms. A couple of the various ones are electroporation, where you shock the cells to allow these giant pores to form in the cell membrane, allowing the protein to go in. You can perform a microinjection by actually physically injecting the cell with the protein CRISPR-Cas9 mechanism. You can do what's known as an Rmp delivery, which allows basically what you do is you modify the Cas9 protein to exhibit a ligand for a receptor that is targeted for endocytosis, so it brings the protein in and then releases it into the cell. Cell penetrating peptide delivery, what you do is you modify the protein to ultimately bring it in as well. And then you have cationic lipid mediated delivery, which is kind of a nanoemulsion where you formulate kind of a lipid sphere around the protein to deliver it in. So I'm going to briefly touch on some CRISPR and Cas9 research related today. It's important to note that currently right now anything that has DNA can be edited by CRISPR-Cas9. We have seen no delineation. It has high specificity to DNA. And it's really divided into two big camps. We have what's known as somatic cell editing, and we have known as what is germline editing. Somatic cell editing is editing the cells of a living organism. Soma coming from the Greek word body. They're part of the organism. When those cells die, they die with the organism. They're not passed on. And honestly, a lot of this research has been very promising. It's difficult when we start talking about editing tissues, but certain diseases or certain genetic disorders like sickle cell anemia, beta thalassemia, and other single nucleotide polymorphisms, such as cystic fibrosis, have been able to be cured potentially by this technology, which is incredible. But where we start to run into some problems is with germline editing. These are germinal cells, so your sperm, your egg, your embryos that are passed on to the next generation, that are ultimately involved in the creation of new organisms. And these traits can be passed on. And the consensus on this type of research is very mixed, because there are a lot of ethical and moral implications that you have to weigh when you decide that you're going to edit an organism that doesn't already exist yet. And so what I want to leave you today with is some actual applications of this technology besides the ones I've mentioned. In the picture on the left, from left to right, we have Dr. Bruce Levine, William Ludwig, Darla Ludwig, and Carl June. Why these people are so important is in 2010, William Ludwig was kind of issued a hopeless prognosis. He had 3 different types of cancer over his body, and he was given only about 6 months to live. But through the work of Carl June and Bruce Levine using CRISPR-Cas9 research, they were able to engineer T cells of his own body that would attack the cancer specifically. It wouldn't attack any other cells, so they cured him in less than 6 weeks using this technology. It was incredible. In the picture in the top right, we have 2 pictures of fish. As you can see, one is very much larger than the other one. They are from the same litter group, but one has been genetically engineered. This salmon, known as the Aqua Advantage salmon, has been genetically engineered with an extra copy of growth hormone, which allows it to grow twice as fast without any of the high nutritional consumption or any issues in terms of, problems in other genetically modified organisms have with feeding them to the population and to market. So that's incredible because it allows fish farmers to not utilize so much space and is allowed to minimize the carbon footprint on the fishing industry, on an environment. And in the bottom right picture, that is of Jamie Galladay. Jamie was born with a condition known as XSCID, or X-linked severe combined immunodeficiency. What this genetic disease basically does is it strips Jamie of his ability to fight infection. He doesn't create any T cells within his body. And this has been characterized as what's known as the bubble boy disease. They have to be kept together, they have to be kept in a very sanitized area, and they aren't allowed to interact with other humans for the fear of them getting a disease and dying. But the Children's Hospital at the University of Cincinnati actually was able to fix this condition in Jamie, providing him with new bone marrow that was edited with CRISPR-Cas9 and now allowing him to live normal life. He's now 6 years old, going to preschool, and as you can imagine, this is real technology. This has real implications, but does have, I would say, a greater moral and ethical implication when we start talking about how we're going to utilize this in the future. So, thank you very much. Are there any questions? Yes?
I have 2 questions. First of all, about the fish.
Yeah.
So, are these fish found in nature?
No, they are not. So they have been specifically regulated to the laboratory right now. And that is one of the big concerns that they have with the fish, is that if that were to be released into the population, what kind of genetic drift do you have? Will that overtake the population and ultimately cause a new breed of salmon? And so yes, that has been a concern. Ultimately, there was such a big outcry about this being a genetically modified fish that it was ultimately removed from market stores like Trader Joe's, Target, Walmart said they would not sell the fish to consumers. So it's been a really big deal, but I would say if done correctly, it could really limit the footprint that fishing leaves on an environment.
But they're so big, they're gonna upset the entire food cycle.
Correct.
And also, what about clinical trials? Those are sold to eat, right?
They would be sold, that was the idea of hopefully having them sold to eat.
So they haven't done clinical trials on if somebody ate a modified one of these, we don't know what that effect would be.
The studies that they have done with that have indicated that there is no difference in between the salmon meat of not a modified salmon and the salmon meat of the modified salmon.
Well, mercury, maybe less mercury.
Maybe less mercury, you are correct about that statement.
Okay, one more thing.
Sure.
This sounds like a very expensive treatment. How soon do you, have you read anything that said how soon it would be available for the general public?
Yes, so it is expensive, and that's one of the major concerns that they're having right now with this type of technology. One of the biggest things that I've read is that they're really trying to, because this is experimentary treatment to a degree, it is going to be expensive at the onset. But what they're hoping is by the time these things start making it to market, that insurance companies will start to see this as a treatment method, just like if you would take a pill to cure a disease, that this would be a potential ability to do it. I'm actually performing some CRISPR research right now for my senior research. It is incredibly easy and effective. One of the biggest things is that the ease of it is allowing it to become more widespread, which can be a good and bad thing. So I would say that in terms of the price right now, yes, it's expensive, yes, it's experimentary, but I think the results do speak for themselves. And if you come back in October, I will talk more about the ethical, moral implications of such technology and how we need to be potentially careful, just like you are trying to bring up, of introducing this into the market system that we have in America.
That was an incredible talk.
Thank you.
I've heard about CRISPR, but I had no idea what it did.
Yes,
You explained that very clear, very good detail.
thank you. Appreciate that. Claire?
I also have two questions. if
Yes.
that's okay. First, so especially in light of this is already happening as you explained.
Correct.
And also with the story that maybe has not been confirmed yet of like the first baby being
Yep.
born, embryo edited with CRISPR-Cas9 technology. So my question is related to the delivery mechanisms that you talked about.
Yeah.
Are there any of those that are more appropriate for something like when it comes to like actual human like embryo editing and some that are not so much?
So one of the delivery mechanisms that I forgot to put up there and forgot to mention is viral injection.
Yeah.
So what you can do is you can place the DNA of CRISPR-Cas9, or you can place the protein component itself into a virus and ultimately have that injected into cells to do that. And that's probably where a lot of the research is being done towards that. But I really do believe that in terms of the delivery mechanisms, obviously you can't shock your body in order to have the cells open up pores. Micro-injection would take a long time. So it really is going to depend on Rmp delivery, the cationic lipid method, and then the cell penetrating peptide chains, these short little chains that are placed onto the CRISPR protein that ultimately allow the kind of facilitate endocytosis of those components into the cell. So does that answer your question?
Very well. And then, sorry.
We can talk about this for
Oh.
hours.
Do you see any, this is totally different type of question, do you see any potential like extreme classism with exactly what she was talking about, some, even more so than we see, it's already an issue, but like some babies being able to just live better lives because they can afford this type of technology?
I'm going to tell you to come back in October.
Is that, yeah.
So I've done some research on that, and I'll tip my hand a little bit. That is a real concern, and just like she was mentioning, it is going to be expensive, at least for the first couple of years. that it is out on the market. And that's where I would say that it is up to scientists, and scientists have actually already done this, to regulate themselves on how far they're going to willing to step. And that's tough. That is really tough. But I would say that progress has been made already. There was a summit in Washington in 2015 formulated by Duna and Charpentier that included over 100 countries of policy makers, bioethicists, theologians, and scientists all coming together on this issue and really discussing. And ultimately, the conclusion they came to is that somatic cell editing can continue, but germline editing, as of right now, with the concerns that we have ethically, morally, but also the ability to do it, and some of the other off-target mutations and mosaicism and all of these other pieces that are added to it, they said right now it's not viable and should not be done. And you can see right now China is cracking down on their CRISPR research right now after that scientist came out with the edits to the two babies. And they are kind of really reining in a lot of the problems with it. So yes, come back in October. I will have more of a formulated thought and an argument on that. Crystal?
Do you think it could be possible, obviously not ethical, but from your understanding of the technology, is it now or could it develop to a place where you could change the personality of a baby before it's born?
Trying to figure out how to answer that. I would say potentially yes, because as we know in biology, our genes are kind of what define us. You can be predisposed to certain genetic conditions like Alzheimer's, like depression, and ultimately those are biochemical reactions that happen inside the body. So could you increase the dopamine receptors and the dopamine production levels of cells? It's very possible, and that would ultimately make you a more optimistic and happy individual. But I would say, do we need everybody to be optimistic and happy? I would say that some cynicism every once in a while is helpful to us to kind of reel back in some broad ideas and generalities. So I don't know if she's cutting me off. She might be. So if you do have any more questions, I'm happy to answer them at the end walking out the door here. So thank you for your time, everybody. Have a great rest of your night.